She hails from Freeman, South Dakota. Her major is biology, her minor is chemistry. And her plans for the future include continuing to be awesome, continuing to be one of the more resourceful students that I have had in my time here at Bethel, and going to medical school at KU. So, Annie?
Well, thank you all so much for coming. I'm going to be presenting on my senior research that I've been working on for almost 2 years now, and it's about variations in cortisol levels across the women's menstrual cycles and the impact of stress on menstruation. So shout out to all the guys in the room. I know you might not want to hear about periods on a Friday night, but I hope you learned something. So historically speaking, research in women's health has been very limited due to the complexities of the women's reproductive system, and this can threaten the quality of medical care that women receive. Often studies contradict each other, and it's a highly politicized topic. So part of my purpose of doing this study was to add to an area of research that has been lacking in the past, and to also do something that applies to half the population. The purpose of my study was to determine if any difference exists in cortisol levels between the follicular and luteal phases of the menstrual cycle, and to provide more evidence on whether menstrual cycle length and regularity are impacted by stress, and also to further discuss the causes and effects of irregular menstruation. So, menstruation is a cyclical process that ranges from 25 to 34 days in length. The mean cycle length is 28 days, and if your cycle is less than 21 days, this is termed polymenorrhea, and if it's greater than 35 days, it's oligomenorrhea. There are two distinct phases that make up the menstrual cycle, the follicular phase and luteal phase, as I mentioned earlier. The follicular phase starts on the first day of your period menses and ends with ovulation, and the luteal phase starts with ovulation and ends when you start your period again. During the follicular phase is when folliculogenesis occurs, which is basically when the female germ cell matures into an egg that can be fertilized. And then during the luteal phase is when the endometrium, which is the lining of the uterus, prepares to receive a fertilized egg. And if it doesn't, then it builds up in thickness to be shed again once the cycle starts over. Here is an illustration of the hormones across the menstrual cycle. You can see, what? Sorry. Well, we're not going to use the pointer. And that's fine. So you can see the 4 different hormones there: FSH, which stands for follicle stimulating hormone, estrogen, LH, which is luteinizing hormone, and progesterone. So you can see the spike before ovulation of estrogen and LH, and then also on the bottom there, the thickness of the endometrium, how it sheds and then builds back up again. So what controls the release of these hormones is what's called the hypothalamic pituitary gonadal axis, which I will be referring to as the HPG axis. So in your brain you have the hypothalamus, and this releases a hormone called gonadotropin releasing hormone. And this hormone communicates to the anterior pituitary gland in your brain, which then releases gonadotropins, which are LH and FSH. And then that communicates to your ovaries to release estrogen and progesterone. Another axis that I'm going to be talking about is the hypothalamic pituitary adrenal axis. And so once again, the hypothalamus and anterior pituitary gland are involved here. The hypothalamus releases corticotropin releasing hormone, CRH. That is communicated to the anterior pituitary gland, which releases adrenocorticotropic hormone, ACTH, and that communicates to the adrenal cortex, which sits on top of your kidneys, to release cortisol, which is your stress hormone. So that's where stress comes in. So here you can see an illustration of these axes I'm talking about, starting with the hypothalamus to the pituitary gland, which cascades down to other glands in your body for different hormone control and release. So the relationship between the HPA and HPG axis is reciprocal. So when your reproductive axis is activated, this increases the activity of ACTH being released, hence more cortisol being released. But interestingly enough, when your stress axis, the HPA axis, is activated for a long period of time, this inhibits the gonadal hormones and inhibits the reproductive hormones that are secreted. So if you have a normal menstrual cycle, this indicates that there is a healthy relationship between your stress axis and your uterus. So cortisol is released when you are stressed, but how does that stressor get received? Starts with the sympathetic division of the autonomic nervous system. And part of the sympathetic nervous system is your limbic system. It processes fear and emotions. And you have the amygdala in there, which receives that a stressor has occurred, which then communicates to the hypothalamus, which controls your physiological response, like increased heart rate, increased respiratory rate, but also it triggers that hormone cascade that I talked about with ACTH and then cortisol release. So cortisol release happens regardless, though, of whether you've encountered a stressor or not. It follows a 24-hour circadian rhythm. So on this graph here, you see how in the morning it is highest, and then at night when you're going to bed is when it's lowest. This is across the board, male, female, you all experience this. So what is the effect of stress on menstruation? Is basically when you are stressed for a prolonged period of time, this HPA axis is continually activated, which increases your cortisol levels, which has been shown to slow your reproductive function and inhibit the release of your gonadal hormones from the hypothalamus. And specifically, then LH release is inhibited, so this doesn't cause that spike of LH right before you ovulate. Studies have shown that in women with higher overall cortisol levels, they can have longer menstrual cycles. It's also associated with dysmenorrhea, which is painful menstruation. Also with general menstrual cycle irregularities, so you can have a really long cycle one time and then next time it'd be short, or then you skip in between there. And interestingly enough, since the start of the pandemic, there has been an increase in menstrual cycle irregularities, which we can contribute to increased stress, whether it be accessing healthcare, financial situations, uncertainty about work, that sort of stuff. So I hypothesized that cortisol concentration would be higher in the follicular phase than in the luteal phase. So when you're on your period versus 2 weeks out. And then my second hypothesis was that in females with abnormal menstrual cycles, which I classified as being less than 24 days long or greater than 32 days long, so on the opposite ends of that mean spectrum, that they would have higher overall cortisol levels in comparison to women with a normal cycle centering on 28 days. I recruited my participants in the spring of 2021 by sending an email out to all the female athletes and concert choir members. And in order to be able to participate, you had to menstruate once a month and track when it should occur. So if you have any long-term birth control that prohibits you from having a period, you could not be in the study. And then once I obtained consent from them, they were enrolled in the study, and upon completion of the study, they got a $5 Mojo's gift card. I had 24 participants. 2 had a dropout, 1 due to scheduling and 1 due to changing up her birth control, so that would provide inaccurate results for me, and they were all between ages 18 and 22. So participants tracked their period last summer for 3 months, so it could be May, June, July, August, however their cycle lined up. And I encouraged them to use a menstruation tracking app to do this so they could try to get notifications and reminders for that. And when they returned in the fall, they reported this data back to me, and using this, I calculated everyone's average cycle length so I could get data on the normal and abnormal cycles. And then when they started their next cycle of menstruating, they reported this to me, and I got their first saliva sample. And then we would schedule up a second time to get the second sample 14 days later at the same time of day due to that circadian rhythm that I talked about earlier. The kits used were passive drool kits from Salometrics. Passive drool basically just eliminates the amount of mucus or possibly food, so you get pure saliva so you could have a better analysis of the saliva for cortisol concentration. And then samples were put in a water bath to make sure there's no SARS particles in there, and then they are frozen until I did my ELISA analysis. ELISA stands for Enzyme-Linked Immunosorbent Assay, and this is a test used to determine the cortisol concentration in each sample. I had to go to WSU to do this because we don't have the correct equipment for it here, and all my reagents and collection aids were purchased from Salometrics as well. So my first hypothesis, you'll recall, was that cortisol levels would be higher in the follicular than luteal phase, and this was not supported. So what you can actually see here of this graph is that the mean concentration was actually lower in the follicular phase than luteal. It was not significantly lower, but the hypothesis was still not supported. You can also see on the wide box plots that there is a lot of variability in the data, a lot of uncertainty in the model, and I'll talk about reasons for this later. I thought it'd be interesting to run another model to compare time of day that samples were collected to see if I could see a reflection of the cortisol circadian rhythm, and that was significant. So in people that I collected in the morning, it was significantly higher than in the afternoon, which matches what would be expected. And then hypothesis 2, in that females with abnormal cycles would have higher concentration across both phases compared to women with normal cycles, was not supported as well. What you can see here is that the mean was higher, but not significantly higher. But most notably here in the model in the box plot, you see the wide margin for abnormal. And I think that's because when we classified abnormal to normal, normal's in the middle of the spectrum and abnormal's on either side. So I think a quadratic model would have worked better for this, but I didn't have the time or knowledge to know how to do that. So for future studies, I could do that. And I thought it was interesting, I required participants to be able to have a period every month to participate. But still, 2 participants did skip a period or 2 in there. So I looked at what their mean cortisol concentration was compared to the rest of the group, and in both phases, they did have higher cortisol. So that indicates that in women with irregular cycles that they do have a higher cortisol concentration, and that stress could be a contributing factor to this. I didn't have a big enough sample size to do any statistical analysis on this though. So outside factors that I think contributed to my results. The big one was anticipatory stress. So my participants will know that spitting into that little tube was difficult and a little bit awkward and embarrassing. So I think the first time when they came in, they didn't know what to expect, but the second time around they might have been nervous, and these nerves and stress could have contributed to the concentration being higher in the luteal phase than in the follicular phase. Another one is that the second sample was collected later on in the school year, so they could have had more academic stress to contribute. And then as far as the second hypothesis goes with tracking abnormal and normal cycles, Females tracked it on their own, and so I have no way of knowing if they were giving me hard and fast data or ballpark estimates, because they were tracking it in the summer, so they might have forgotten sometimes and just had to guess. Also, with them tracking it over the summer versus during college, summer's not as stressful of a time as during college, so for future studies I'd be interested in tracking over during the school year, maybe like a whole year, so I could get a more representative sample, and then also tracking over multiple periods as well. And then my sample size was small, and once again, length of study. I only got to get saliva samples from one period, and it would have been interesting to see over multiple how it compared. So a big thing that we hear about a lot as females is premenstrual symptoms, PMS, and this happens in the late luteal phase and stops before you, or like shortly after you start your period. And this is described as physiological, psychological, and behavioral changes that cause distress and disturb everyday functioning. And a lot of previous studies that I looked at had women reporting increased feelings of depression, anxiety, just general negative mood during this phase. So I'd be curious if I would take cortisol samples in more than just 2 times across the menstrual cycle, but during multiple phases, including the PMS phase, how that would affect and what we would see in the cortisol rhythm throughout the entire cycle. So I want to talk a little bit about the impact of irregular menstrual cycles on women's health. So there are some factors that are typically associated with irregular menstruation, that being BMI, smoking, physical activity, and other lifestyle choices like the amount of sleep that you're getting, that sort of stuff. But the biggest factor is HPA axis dysregulation. So when you have the hormone imbalances due to outside stressors or simply that mechanism releasing the gonadotropin releasing hormones is not functioning correctly, you get these hormone imbalances, which causes stress and prolonged activation of the HPA axis. And this eventually leads to your immune system being suppressed and chronic diseases, because there are glucocorticoid receptors on every major tissue and organ system of the body. So it can affect everything, including your reproductive system, which these chronic diseases can eventually be like type 2 diabetes, cardiovascular disease, osteoporosis, and infertility. So in conclusion, it's important to mitigate any chronic stressors that females are experiencing that can lead to this dysregulation of the HPA axis, especially in college when it's already a stressful time for many. And hopefully by doing so, fewer females can experience hormonal changes that are leading to irregular menstruation and then eventually reproductive health concerns and chronic diseases later on in their life. So I'd like to thank my advisor, Dr. Francisca Mendez-Harklerode. She has been an amazing help and support throughout this. A big shout out to my research assistants, Claire Brocksterman. She's helped me so much, and she's just a freshman. She's so smart. Sandy Dow, she came with me to WSU to help me with the ELISA analysis when I didn't know what I was doing. And then Mickey and Katie, they helped me for a semester with meeting with participants, reading my paper. Dr. William White, who I mentioned, helped me at WSU. Brad Celestin and Brenda Haynes helped me a little bit with the statistics. The URICA committee, I got grant money from them, which I used to purchase collection kits and the gift cards for my participants. My mom, she's read my paper multiple times, so it doesn't have grammatical errors that Fran has to deal with. Walter Ring was the person we worked with from Salometrics to get all our correct kits, and he was very helpful. And then Jared, who helped me organize this tonight so I could present to you all. Here are my references. And I think we have time for a couple questions, but in the interest of staying on time, like 1 or 2, and you can talk to me afterwards.